Good morning, good afternoon, and good evening, everyone. This is Liam Sanyo from Inside Scientific, and I'm very pleased to be your host for today's event, which is titled EEG Monitoring Approaches to Predict Learning and Memory Changes in Early Alzheimer's Disease. This webinar has been sponsored by DSI, a division of Harvard Bioscience, so a big thanks to them for making this event possible. Today, we're being joined by Dr. Fiona Harrison, Associate Professor of Medicine at the Vanderbilt University Medical Center. Dr. Harrison will discuss how vitamin C deficiency and exposure to toxins can impact glutamate uptake and clearance, and how even small changes in neural signaling can be detected by monitoring EEG activity and correlated with performance in learning and memory tasks. And with that, I'm very pleased to introduce Dr. Fiona Harrison. Fiona, thanks so much for joining us today, and the floor is yours whenever you're ready.
Well, thank you. Thank you for that introduction, Liam. I'm delighted to be here today to share some of the recent work from my group, trying to better understand how changes in epileptiform activity may be associated with cognitive decline in Alzheimer's disease and other related dementias. So my lab is primarily interested in lifestyle differences such as diet, environmental exposures to toxins, and other illnesses or comorbidities that can impact the trajectory of Alzheimer's disease. So while I'm going to start with a reasonably general story, toward the end of the talk, we will get into some new data to help explain this image that I have here of the mouse in the orange. So I think everyone here is already aware of what a huge and growing problem Alzheimer's disease is in developed countries such as the US, but rates are increasing worldwide. As we get better at curing other diseases, more people live long enough to develop AD. But, we may recover from these other conditions without necessarily staying healthier. So there are many more cofactors increasing rates of diagnosis and lowering the age of onset. So these data from the Alzheimer's Association infographic predict something like 1 in 3 people born today will go on to develop some form of dementia as they age. Now, we typically think of Alzheimer's as a disease of aging, but in many ways, it's the culmination of a lifetime's brain health and should perhaps therefore be studied and treated accordingly. So Alzheimer's disease is defined on the presence of beta amyloid plaques in the extracellular space, hyperphosphorylated tau tangles within the neurons, and cell loss causing overall shrinkage of the brain parenchyma with increasing ventricle size. The recent controversy regarding the approval of the beta amyloid plaque-removing drug aducanumab, or Aduhelm, has really reignited the conversation around the amyloid hypothesis and whether this should still be the focus of so much research. I'm sure everyone here is familiar with that case, but a very simplified summary is that the drug was effective in clearing amyloid plaques in many of the patients, but the risks of side effects, including ARIA, microbleeds, was high, required careful monitoring during the study, and that's just not feasible in most patient populations. And any cognitive benefits were mild at best and only in a limited number of participants. So this leaves the question, how important amyloid plaques really are in cognitive decline. But one of the interesting and in fact really hopeful facets of Alzheimer's is that there is a huge difference in the clinical manifestation, so the dementia and other behavioral changes that patients experience in relation to the pathology within the brain. So we often see comparable brain atrophy in cognitively normal and impaired adults. And to illustrate this, in this data set from several years ago, we have 137 adults imaged for amyloid, and you can see, a large spread across the population, including some high amyloid in the older populations. But the key feature here is that all of these participants were cognitively normal when this study was performed. And interestingly, we can also see amyloid is detectable in much younger adults, even if it is at the lower levels detected. Still, many of the major pathologies known in Alzheimer's are both triggered by amyloid and tau pathology and also serve to accelerate those changes. For example, in the case of microglial response to amyloid, you have a need for activation to clear amyloid, but then that leads to long-term activation, which triggers damage when microglia destroy healthy synapses instead. So it becomes more important to ask how the brain is responding to each of the pathological signs rather than measuring frank levels per se. So in mice, using well-known transgenic models, over the past decade and a half, many groups have observed clear associations between amyloid deposition and behavior. So in the first example here, CRND8 transgenic mice between 3 and 12 months of age were trained in a conditioned fear task, and cognitive deficits increased with age, in line with the age-related amyloid buildup. In this second example, we have performance in 16-month-old TG2576 mice that was assessed using an alternating T-maze task. And again, performance was correlated with amyloid deposition. But if these relationships are not true in human populations, then how do we get from the basic pathology to cognitive decline? Now, there are multiple contributing risk factors to cognitive decline that connect the extent of pathological damage observed to the experience of cognitive, emotional, and other behavioral changes experienced by patients. So several of those are listed here, and nutrition here includes both undernutrition and specific dietary deficiencies, as well as overnutrition through high-fat diets and the metabolic syndrome and type 2 diabetes that are related to that. Exercise, general activity levels, lifetime health history, and continued intellectual and social engagement are all known predictors of an AD diagnosis. We're also now gaining much more information about the importance of sleep in Alzheimer's pathology, as well as the possibility of roles of specific environmental toxins. And there are several key areas or themes under investigation across numerous labs right now, with focuses on specific pathways linking these variables and individual differences with amyloid and tau pathology and with clinical memory loss. So each of these pathways that I have listed here has been studied extensively, and yet we're still searching for common links among them, as well as looking for the most predictive biomarkers that could be of use in human populations. To me, the really encouraging thing about Alzheimer's research is that there is so much we can do in our lives to prevent the cognitive decline, or at least slow its onset even in the face of pathology. And my lab is currently focused on asking how environmental and dietary factors influence one particular facet of the related pathology. And that's the excitotoxicity associated with changes in glutamatergic signaling. But although I am going to focus there today, I will state right now that these changes do not happen in a silo. And all of these other factors listed on the slide are also involved, particularly in this case, neuroinflammation and oxidative damage. So, what are the factors that can determine the relationship between an observable pathology and clinical presentation of cognitive and other behavioral symptoms? So the primary effect of alterations to the glutamate system is, hyperexcitability, and in case of, even greater disruption, ultimately leading to seizures. And, by this, I mean cells are firing out of turn with disrupted or asynchronous rhythms. This can be measured at the single cell level in animal studies and at a more global level using EEG in animals and in people. And it's now well known from both animal models of Alzheimer's and observational studies in clinical populations that there is a strong relationship between seizures in Alzheimer's and cognitive decline. But it's a complicated story for which many questions still remain, particularly when those seizures are silent. So epilepsy or subclinical epileptiform activity appears to accelerate onset of cognitive decline by 5 or more years compared to non-epileptic controls. And in the study I've cited up here by Keith Vossel et al., epileptiform activity was observed in 41% of these were non-Alzheimer but mild cognitive impairment patients, and in more than 50% of those cases, the seizures were non-convulsive. So abnormal epileptiform activity without visible convulsions can easily go unrecognized and untreated, and may reflect underlying pathologies. To add to the challenge of diagnosing this in patients, such changes may not even be visible on surface EEG, and so may require implanted electrodes to fully understand the patterns of altered excitability. In addition, the greatest changes in epileptiform signaling observed between control and AD populations, and this is in both humans and animal studies, are usually typically observed during sleep, which can further complicate diagnosis. So putting all of this together, it seems that altered epileptiform activity may be both a symptom, but also a driver of cognitive decline. Now, one of the reasons I think that we can have quite good confidence in this line of research is the strong resemblance of the animal models to the clinical phenotypes. And that is not always the case, particularly in the Alzheimer mouse models. But it actually turns out that almost all transgenic mouse models of Alzheimer's have EEG or seizure abnormalities. And importantly, it seems that this may be a much more translatable phenotype between the animal models and human disease. And so there is, in the paper I have cited here, there's a list of, it's not an exclusive list, but there's a list of many of the models that do show changes in EEG and epileptiform activity. So in this first example here, for one of the mouse models, we have EEG recorded in anesthetized APP-PSEN1 mice for 15-minute recordings. The most frequently observed abnormalities in the transgenic mice were the interictal spikes, and those were seen in animals as young as 4 months. In the second study, we see recordings in TG2576 mice that were begun at just 5 weeks of age, and the transient spike-like activity, you can see that in the left-hand panel marked with the black arrow. Those were observed in all of the TG2576 mice studied, but none of the wild-type glutamates. And mostly those were observed during REM sleep, which fits with the human profile that we observe. The authors also noted that those effects were observed prior to amyloid deposition in this model, but of course that doesn't necessarily mean prior to any amyloidogenic changes. There would still be generation and some accumulation even at that age. So in my lab, we wanted to test the hypothesis that even small disruptions in epileptiform activity would be sufficient to induce or accelerate cognitive decline. And we did this in APP-PSEN1 mice, which are highly susceptible to seizures and EEG abnormalities anyway, particularly as they get older. But we performed our study in young adult mice between 3 and 6 months of age, in which actual plaque deposition is very sparse, particularly when these mice are on the C57 Black 6J background, which is what we use. I'm going to present 3 different experiments that we used in this study, and although the specific details of treatments vary for each individual experiment, the overall design is the same. I do need to state that these data are currently unpublished. We actually have submitted our second round of revisions in response to some really excellent discussion with the reviewers, and we are optimistic that this will be in press soon, and when that happens, we can add it to the resources site that you have access to here. So kainic acid is a glutamate analog. It's an agonist for the kainate subtype of ionotropic glutamate receptors, and those have the highest density in hippocampus. The amygdala and various cortical areas also express high levels of the kainate receptor, so these are the areas preferentially affected or damaged after systemic administration of kainic acid. And that's why, we thought this would be a useful tool for an Alzheimer's disease-related study, because these are the areas that are most highly impacted by pathological change. Now, high doses, high concentrations of kainic acid do induce seizures, also cell death and mortality, but we were very careful, or we tried to be very careful to use much lower doses in our studies. We used a repeated dosing strategy in order to model chronic hyperexcitability from young ages. And in each of the data sets that I'm going to present, the data should look the same. So wild-type mice will always be in white, transgenic APP-PSEN1 mice will be in a darker shaded color, and then control or saline-treated animals are in black and white, and then any color denotes the experimentally treated groups. So in our first experiment, we were helped by Dr. William Nobis. He's an assistant professor in neurology here at VUMC. We took 12-week-old wild-type and APP-PSEN1 mice and treated them for 5 consecutive days with 5 mg/kg kainic acid. So that's a very low dose. It does not induce observable seizures in the animals. In fact, you can barely see any behavioral signs of the treatment at all. So as you all know, I'm sure, long-term potentiation, or LTP, is the process of strengthening connections between synapses in response to neural activity. And deficits in LTP are therefore often considered a molecular marker of learning and memory ability. And the field excitatory postsynaptic field potentials, or FEPSPs, were recorded from the CA1 region of the hippocampus by stimulating Schaefer collaterals in the stratum radiatum, and that's the image on the top right there for a schematic of how we did the LTP. So all of the baseline and control measures that were made were really similar among the groups. but kynic acid caused a decrease in the post-tetanic potentiation period around 5 minutes after stimulation, but those mice had normal appearing late phase LTP. And interestingly, that pattern of kynic acid in wild-type mice looked very much like the saline-treated APP-PSEM1 mice. That's something we had noted before and suggests these changes may be related to alterations in presynaptic release of glutamate, potentially postsynaptic clearance due to the acute kynic acid injection. The similarity between those kynic acid-treated wild-type mice and the saline-treated APP-PSEM1 animals also helped to confirm to us that this type of stimulation protocol was, appropriate for our experiments. But in contrast, I think you can see in that red line underneath the other main body of the data, the APP-PSEM1 mice that received kynic acid showed really robust impairments in hippocampal LTP. And that suggests to us that potential impairments in postsynaptic glutamate clearance, and also clearly highlights the increased sensitivity to the drug in those mice. We were therefore reasonably confident that our treatments might do something to the learning and memory abilities of the animals. These recordings were made within 2 hours of the final treatment, so this is catching them in an acute disruption period. We did not look at the mice after washout of the drug as well to see if those effects were persisting. Instead, we moved to look at what any behavioral effects of those treatments might be, since that's really the main focus of the lab in these studies. So in the second experiment, we started with mice at the same age, 12 weeks, but this time we treated them with 10 mg per kg kynic acid, and we did that twice a week. We thought we would be safer with this slightly higher dose because it still does not cause, noticeable behavioral responses or seizure responses, and these treatments were much more spaced out than in the LTP study. Unfortunately, you can see from the panel on the right that despite this low dose, we lost about 80% of the kynic acid-treated APP-PSEM1 mice. And I will say, in no case did we observe seizures immediately following treatment. The mice died, presumably due to spontaneous seizures in the home cage, usually occurring at night during the active phase, so we would find those mice in the mornings. So that meant that after 3 cohorts of mice, we were forced to add a 4th cohort in which we treated animals with an even lower dose, 7.5 mg per kg, in order to provide sufficient power for our studies. Now, we always perform a wide range of tasks, including controls for activity, anxiety, and motor ability. I'm only going to show the key behaviors today as I go through this data set. So first of all, we use the water maze. We train the mice to find the hidden platform in this water maze task. And as you would expect in young 5-month-old mice, even in the transgenic animals, they were all able to perform this task. So they all showed decreased escape latency over time. They were also able to show preferential swimming in the target quadrant during the no platform probe trial. So you can see the left-hand bar in each of those groups of 4. The diagonally striped bar is much higher than the other 3 quadrants. And then those heat maps on the right are some examples showing preferential swimming time around where that platform was located. So one good thing about the water maze is that we are able to make it a little more challenging when necessary. So we included reversal learning, and in this, the platform is moved to a new location in the pool, although nothing else about the room has changed. And this is actually a lot harder for the mice because they need to not only learn a new platform position, but also remember to forget the old platform position. And although all of our mice were able to acquire the task, which you can see there on the left, again with decreasing escape latencies, memory for the new location was poorer, but only in the kynic acid-treated APP-PSEM1 mice. So you can see that in the top right-hand panel. So again, the target quadrant is marked with the diagonal lines. This time it's the far right bar, and you can see each of the first 3 groups are able to show preferential swim times, which the kynic acid-treated transgenic mice did not. Now, this may seem like a trivial difference, and in many ways, all of the behaviors I'm going to present today are quite subtle. But given these mice were young, otherwise healthy, and in particular, these were the surviving mice in this study. So by definition, they are the least affected in the group. To see anything here, at this age, after these mild challenges, is actually quite exciting to us and indicates just how early in the disease process these kinds of cognitive changes may be occurring. And then to give you an idea of how subtle this post-kynic acid behavior is, I'm going to show you some data using these force plate actimeters from BASI. So the software output there in the yellow box in the middle looks very much like an EEG. In fact, though, this is picking up movement using very sensitive force meters at each corner of the chamber. So the three spikes you can see in the top middle panel are actually myoclonic jerks. So we can confirm those using paired video recordings. But this equipment is sensitive enough not just to detect movements like that, but to tell the difference between a mouse with low activity who might be grooming or sniffing in a corner versus a mouse that is actually frozen, as is often observed following treatment with kynic acid or other seizure-inducing drugs. So you can see from these data here, which show the total bouts of low mobility, that APP-PSEN1 mice are more likely to exhibit the real immobility than wild-type mice following treatment with kynic acid. It's really quite difficult to pick up with the naked eye, but this is what led us to now look at the EEG data to get a stronger picture of what was really happening in the brains of these mice. And you'll also notice that data set there on the bottom right is just 20 minutes following treatment, and only during about 10 minutes of that do we detect a significant difference. So again, at these low doses, we're looking at a very short acute effect of drug. So here is where we get to the EEG part of the data. And to complete these studies, we use the DSI system, which has a number of advantages for our study design. So for those of you who are not familiar, instead of a traditional head-mount tethered system, the DSI telemeter implants are surgically placed just above the hip, and the wiring is tunneled under the skin so that the EEG wires are placed in holes drilled in the skull, touching the surface of the brain, just like with any other EEG setup. The EMG wires are secured in the nuchal muscles at the back of the neck. Nothing is protruding. The mice heal really nicely, and they can be maintained for long periods of time with the telemeters in place. They're turned on and off just by waving a magnet near them, and that allows for really long-term recordings if needed. So the example in the image below shows paired video data with the EEG. This is in the mouse, a mouse in the middle of a seizure event. It's actually not from this study. Again, we were looking for mice without these seizure events. You can see, hopefully, behind the food cup, there is a mouse in the characteristic sort of rigid posture with a tail extension. He's hiding, so the yellow there marks the head, the back, and the feet, and then the red line is the tail extension. So perhaps you can see him hiding there, in the midst of a seizure. The other benefit of the system is that although the mice are a little bit lumpy by the time we finish with them, there are no other impairments to movement really, and that allows us to complete behavioral testing in the same set of animals. And that's what we did here in this study. So we did 4 weeks of EEG recordings, during which time the mice received those twice per week kynic acid treatments. This time we had learned our lesson and we went back down to 5 mg per kg, again looking at very small changes, small but chronic changes. And then following those recordings, the mice underwent a week or so of some behavioral testing so that we could get measures from within the same animals that we had EEG recordings. So first of all, in line with the human data I described earlier, we're not looking for actual convulsive seizures. We did observe seizures in one APP-PSEN1 female and one wild-type female treated with kynic acid during the study. Those mice were removed from all of the analyses. 2 male APP-PSEN1 treated with kynic acid died during the study, presumably from seizures. We only caught one of those on the EEG system, and so all data from both of those mice were removed as well. So again, we're trying to catch the animals with what could be termed clinically silent changes, and again, which are the least impacted in the group. So in this first observation, we counted the number of spikes or spike trains, polyspikes, which had no associated behavioral signal either by EMG or by the paired video. And you can see those in the middle panel on the left, highlighted by the green bar and by the arrow. We also saw some spikes that did have a small EMG signal associated. These are confirmed on video as a small head bob behavior, and that's, representing a small myoclonic jerk. But as you can see at this age, there's very little difference between the wild type and the APP-PSEM1 mice at baseline or under saline treatment. Following chinic acid treatment, much like with the LTP and behavioral studies, there's a far greater response in the APP-PSEM1 mice, and those were significant increases. It's just not marked on the panels there. So these specific signals are all automatically detected using the NeuroScore program, but we do go through by hand to confirm all of the markers placed by the program. We also remove any instances of signal dropouts. And of course, we just want to get a stronger picture of what is happening in each mice. So we do a lot of hand coding of these data as well. Afterwards. We also use the NeuroScore automated analyses to help with parsing out the data according to the frequency of EEG signal. Now, data from human studies show changes in delta, theta, and alpha frequency signals, and the magnitude of those changes may well be linked to the fact that the biggest changes observed in many studies are during sleep, both in REM and non-REM sleep, and those are mostly registered as delta or theta. We also use the NeuroScore data and guide parameters provided by DSI to confirm patterns of sleep and wake, and found very little difference between the genotypes, at this age. So these recordings were all made during a 4-hour period during the light cycle, when mice should mostly be sleeping, which you can see that they are, although about 30% of this time is classed as awake. We were able to perform these studies in a dedicated housing room. So other than daily checks by the veterinary staff, which typically occur in the mornings, or when investigators, from our lab were in the room, there is really very little to disturb the mice. We even have the computers outside of the room so that they aren't bothered by that noise either. And although we did observe genotype differences in the different power bands, those are marked by the asterisks, and you can see we have the data broken out into REM sleep, non-REM, and wakeful periods. We didn't pull out any, any clear additional effects of the chinic acid treatment. We're also not able to pull out specific changes in the delta frequencies, which was where, we had predicted the most changes would be. But this indicated to us that the spikes we observed were perhaps, a discrete and specific marker of change, not related to overall EEG readouts or overall disruption in sleep patterns. And so they might have some predictive value. So as I mentioned, one benefit of the DSI system is that we can perform behavior in the same mice that we have the EEG recordings from. Something like the water maze is perhaps not ideal, since the implants might impair swimming. So instead, we focused on fear conditioning. During the initial trial, our mice are trained to associate a 30-second tone with a 0.5 milliamp shock that co-terminates with the tone, and we use 3 tone shock pairings across an 8-minute trial during our training paradigm. So 24 hours later, mice were placed back in the same testing context, and we look at freezing behavior to assess memory for the shock. So all of the mice, again, learned this association. You can see that from the increased freezing when placed back in the context compared to baseline freezing behavior. That's in the top left-hand panel. And that's as you might expect, again, in young animals. Even in the transgenics at this age, we don't typically see a lot of cognitive deficits. So as before with the water maze data, it was not until we used the slightly harder aspect of this trial, of this task, and that's the response to the cue, which was the tone in a non-familiar context, that we saw a deficit emerge in the chinic acid APP-PSEM1 treated mice. So this part of the task is thought to be more dependent on intact amygdala function rather than just hippocampal-based learning. There are kainate receptors in both areas, but it's interesting to us to think that the circuitry involved here may not just be cortico-hippocampal, which is often what's discussed in these data sets. So what's causing these effects? Well, downregulation of EAAT2, also known as GLT1 protein on astrocytes, is associated with elevated glutamate levels and neurotoxicity. Since this is the major protein responsible for clearing glutamate into astrocytes, this is perhaps not surprising. Several post-mortem studies indicate that GLT1 levels are decreased during Alzheimer's in concert with increased astrocyte activation. And so that's picked up in this case on the right by increased GFAP staining or Western blot at the same time as they have decreased GLT1. So I'm showing here our data now, Western blot data from the cortex, since that's where our EEG recordings were made. But we also saw similar response to this in hippocampal tissue. So at this age, by Western blot, we did not have any changes in GFAP levels, suggesting that the mice are relatively spared from gross neuroinflammation at this point. And that's, likely sensitive and responsive to amyloid levels as those build up over time at later ages. We did, however, see decreased GLT1 expression in both wild-type and APP-PSEN1 mice in response to kainic acid, with a cumulative effect in the kainic acid-treated transgenic animals. So there's reason to believe that our treatments were having some direct and lasting impact on glutamate homeostasis, and that may be important in the behavioral outputs that we saw. So I hope that those experiments together convinced you that even very mild perturbations in glutamate signaling can impact cognition, and that this can be observed through changes in EEG, even in very young animals and at very early stages of disease. And given this, it becomes particularly important to identify alternate triggers of glutamatergic dysfunction and hyperexcitability. So here are just a few examples. Each of these are associated with electrographic non-convulsive seizures and increased likelihood of an Alzheimer's disease diagnosis. So air pollution, there's a lot of factors in air pollution. I think this image is Mexico City, but manganese is one of the factors that's involved there. We also study manganese in the diet in my lab, and there are several neurotoxic effects, particularly in cases where people might obtain water from wells. We have increased manganese. People exposed during workplace exposures as well. Glutamine synthetase is a manganese-dependent enzyme critical in glutamate recycling, but in excess manganese is neurotoxic and can therefore impact glutamatergic function through other oxidative stress mechanisms as well. There are other interesting stories recently in the literature involving direct triggering of glutamatergic systems, contributing to acute onset of cognitive decline and seizures, so in the case of domoic acid, or more long-term changes in the case of the flying foxes up there. We're studying the roles of manganese right now in my lab, and additional studies also considering the roles of repeated mild traumatic brain injuries and acute illness, including delirium, on development of Alzheimer's disease. And we're using EEG as a tool in all of these projects. We hope to be able to share data from those studies over the next 12 months. But the first three examples, whether direct neurotoxins or not, are actually all also acquired through diet, whether on purpose or accidentally. And the interesting thing about dietary studies is that sometimes we study excess toxins and sometimes we study deficiency. And, this talk is about environmental influences on EEG as well as Alzheimer's development. And one dietary factor I have always enjoyed working with is vitamin C, or ascorbic acid, and its roles as a specific neuromodulator as well as a powerful antioxidant. So most people are aware of the condition of extreme ascorbic acid deficiency, that's scurvy. And scurvy is rare, it's not unheard of. That's actually not what we study. We study depletion, and deficiency that is not scurvy. And those are actually quite common, particularly in older populations at risk of Alzheimer's disease. And one reason that humans are susceptible to vitamin C deficiency is because, along with other primates, with guinea pigs and fruit bats, we have a non-functional copy of the enzyme galonolactone oxidase that's required for vitamin C synthesis in the liver. So all other animals, importantly, including, lab rodents, rats and mice, make their own vitamin C and therefore can never become deficient. So for humans, for us, it's not hard to get sufficient vitamin C in the diet. But even at recommended dietary intake levels, it's also reasonably easy to have less than replete levels. So you can see marked in the first smaller red box, if you take the recommended daily intake level of between 45 and 120 milligrams per day, and that depends on age, sex, pregnancy status, and in fact, different countries have different guidelines. Those amounts are not going to get you to a replete status. You need more like 250 or more mg per day to get you there. And the reason that vitamin C is particularly relevant to the current story relates directly to glutamate transport. So as glutamate is released into the synapse, it binds NMDA or AMPA receptors and calcium influxes into the cell. Then it needs to be efficiently cleared from the synapse before it can become excitotoxic. And around 90% of the glutamate in the synaptic cleft is cleared through GLT1 on the astrocyte. As glutamate is taken up, the astrocyte swells, and that leads to opening of these volume-regulated anion channels. And that allows ascorbic acid to be released into the synapse at exactly the time that glutamate is present and additional antioxidant protection might be required. It's then also available for uptake by neurons on the SVCT2, which is the sodium-dependent vitamin C transporter. And so if the neurons are under stress, they can increase their antioxidant protection that way. Following donation of electrons to neutralize any reactive oxygen species in the synapse, ascorbic acid in the extracellular fluid becomes oxidized to dehydroascorbic acid, that's DHA. That's then taken up on glucose transporters in the astrocyte because, while neurons have the SVCT2, astrocytes do not, so they take up the oxidized version of ascorbate. Within the astrocyte, the DHA is then reduced back to ascorbate or vitamin C, which is then available for release again. And so these two processes of glutamate clearance and ascorbate release are really intimately tethered. And it used to be deemed a hetero exchange, although that does not fully explain the relationship. Stimulating cells or applying glutamate absolutely causes ascorbate release, that's clear, but how or whether ascorbate level itself impacts glutamate uptake is less clear. But if ascorbate is not available, then you have potential for damage to neurons, increased oxidative stress imbalance at the synapse, and potential post-translational modifications of transporters like GLT1, which is sensitive to oxidative stress. So how did we go about testing the hypothesis that low ascorbate was interfering with glutamate signaling? First thing we did, we took gulo knockout mice. So they are like us. They cannot synthesize their own ascorbic acid. So they have to receive it from the diet. And we kept them on a high wild type equivalent vitamin C level or on low levels for 1 month. And the levels that we kept them on are not enough to induce scurvy. They give them about a 50% decrease in brain vitamin C levels, but there's no ill health, no weight loss, nothing like that. And you can see in the panel on the left that under these lower vitamin C conditions, mice were far more sensitive to the kynic acid with a faster onset latency to that first head bob behavior. And many of the wild-type mice never show that behavior. Again, 10 mg per kg kynic acid is not a large dose in otherwise healthy mice. And our contention is that this kind of depletion is possible in humans because this is not an unhealthy level that would send you to the doctor, but it's a poor diet or a smoker that uses up the rest of their vitamin C stores. We tried to look at this a little more closely in SVCT2 heterozygous knockout mice. So these mice are lacking 50% of that specific vitamin C transporter in the neurons. That leads to about a 20 to 30% decrease in brain ascorbate, so not as large a difference, even though those mice can synthesize their own ascorbate in the liver. These data required using a Pinnacle system that has a mouse tethered to the receiving device, and ideally is used for only shorter term measurements of around 24 hours in order to support mouse well-being. So although we saw that both knockout of SVCT2 and APPPSM1 genotype alone, slightly increased the spikes observed in the animals following kynic acid, the largest differences we observed were only seen in the combination case of decreased ascorbic acid and APPPSM1 mutations. And that suggests to us that there's some effect of low vitamin C accelerating the deficits observed according to the AD genotype. So the nice thing about the untethered EEG system that we use now is that you can plan really longitudinal measurements. And this is handy for a number of environmental or pharmacological based studies. In this study here, and I have to caution that this is still ongoing, has not undergone any peer review at this point. We're still actually adding a few more animals in. We horribly lost one cohort of animals during the beginning of the COVID shutdown. We had to close out that study quickly, so we are a little behind in finishing up this data set. But this is testing the effect of chronic vitamin C depletion with no additional challenge, so in this case no kynic acid, in both young and aged APPPSM1 mice. And based on all of our other studies with the kynic acid, we hypothesized that the lower the brain vitamin C levels became, the more of the aberrant spike activity we would see. And we used a within subjects design in which ascorbate levels were slowly decreased in all of the mice, which we hoped would be more powerful given the likelihood of greater variability in these older mice. We've got 4.5-month-old mice and 18 to 19-month-old mice in this study. And all of these mice are gulo knockouts, so are dependent on dietary vitamin C, and then half of them also have the APPPSM1 mutations. So in the first week, we actually did see what we were expecting. We have approaching significance here, even with just 3 or 4 mice in each group. You can see also the expected effect of increased spikes according to genotype. In the older mice, you can see more spike activity at baseline, both the wild types, particularly the APPPSM1 mice, and also any potential effect there of the vitamin C depletion is greater in the APPPSM1 mice. But of course, nothing is ever as simple as we would like. And over consecutive weeks of depletion, we observed no further increase in any of the groups. And in the aged mice, we saw what looked like a recovery of this activity. So I do not want to suggest at all that decreased dietary vitamin C could be protective of brain health. We have a lot of information to show that this is not true. So we definitely wanted to study this phenomenon a little more closely. But what is clear here is that there is a complicated story that requires longitudinal measurements for us to fully understand. Taking a snapshot approach in this kind of study would obviously lose us an awful lot of data. So as one additional marker, we used the root mean square of all the overall signal. It's a calculation that loosely provides a measure of the overall power or activity in the signal. This should be reasonably stable, but is generally depleting over time in the aged mice, and so may reflect some form of lower engagement or even neural fatigue in the cortex. We have not fully interpreted these data yet. It's interesting to us that there is a generalized difference in the data that we observe in the aged mice over time that we do not see in the younger mice. If anything, the younger mice are working harder, with an increased signal over time. So as we complete our final cohort of animals and add in the extra data, we will be using multiple different analyses through the NeuroScore program to better understand what all of these data mean. So finally, in these animals, at 12 months now, representing an in-between time point for the 2 age groups used in that EEG study, we see at this time, we do have astrogliosis reflected by the increased GFAP in the APPPSM1 mice. Because of this, we normalized to GFAP rather than just actin. And we see here the expected decrease in GLT1 according to genotype, so that's pretty clear. But again, there's no difference according to our other experimental manipulation of ascorbate level. And that suggests that whatever these changes are, whatever they're caused by, it's not as simple as just glutamate clearance. So in future, we'll be combining these studies looking at glutamate release under different triggered conditions using in vivo sensors such as iGlu sniffer. We have some GLT1 conditional knockout mice that we will use to probe specific, the role of specific brain areas. And there are, of course, multiple other contributing mechanisms as described earlier, and many of these are specifically implicated with roles of ascorbic acid, including dopaminergic function, cholinergic changes, oxidative stress mechanisms, mitochondrial damage, and many more. So to conclude, increasing amounts of data from animal and human studies show that subclinical and non-convulsive seizure activity are an early part of Alzheimer's pathophysiology, and they may be sufficient to trigger cognitive decline in some groups. Specific EEG markers could therefore be a useful early predictor of later cognitive decline, and in fact help identify specific subgroups that may be amenable to preventative pharmacological interventions. Diet and environmental exposures may contribute to cumulative damage throughout life, and if we can better understand these roles and how to manipulate them to our benefit, this could also provide new insights on possible interventions. With that, I would like to acknowledge the funding bodies that have supported this work, both at the NIH and the VA, but even more importantly, I want to acknowledge my outstanding colleagues and lab members. So Dr. Bill Nobis performed the LTP studies for us and continues to support and train folks in my lab to perform the electrophysiological studies. He is also a neurologist who reads real human EEGs, not just mouse data, so has been a great help in interpreting these data as well. The EEG analyses themselves were all performed by graduate students, Rebecca Buchanan at the bottom and David Consoli, and by postdoctoral fellow Dr. Jordan Wilcox. And so with that, thank you all for your attention, and I am happy to take some questions.
Fantastic. Well, thank you so much, Dr. Harrison, for the really fantastic presentation and for sharing your work with us. This is a really interesting avenue for research. All right, so let's jump in. First question, so given this possible direct link between the increasing abnormal epileptiform activity and memory impairments, are there any behaviors that are likely to be particularly sensitive to seizure phenotypes or to kyanic acid if tested experimentally?
Yeah, so there are studies in patients that were cited earlier in the talk that used a number of complex measures, a lot of different test batteries, and they found sensitivity to epilepsy in some of those outputs, but not others. Of course, many of those tasks like clock drawing or, serial subtractions are not necessarily relevant to everyday life in patients. But this does tend to suggest that different pathways or circuits are impacted. So in mice, since we think that the epilepsy is driving the same pathways that are impacted early in disease, I think that many of the classic tests of learning and memory that are dependent on intact corticohippocampal circuits, are implicated. So really anything that your mouse model has deficits in any way might be accelerated by the additional epilepsy phenotype. So for us, testing in young mice, the most important thing is probably not the specific task used, but that all of the correct control tasks are included. So we're controlling for activity and anxiety and motor deficits, for example. That helps us interpret the final, the most important output that we're interested in. And because we were testing in young mice that really had pretty intact performance on most of these tasks, it's easy to get a ceiling effect and to not see your deficits. So it's essential to have tasks that you can adapt the protocols for in order to make harder. So for us, that was reversal learning in the water maze using cued and context-dependent tasks and conditioned fear. Other tasks people use, you might want to, decrease the amount of training or increase test delays, for example. Anything that's going to prevent that ceiling effect and allow you to detect differences between your groups.
Fantastic, great answer. Here's a great question here. So you were saying that the EEG data reported in the APP-PSEN1 mice were made during the day, which is the light phase for the mice. Do you think you would have expected different results if you'd done your testing during their more active dark phase?
Yeah, I do think we would see some different results, not not different enough to lead to alternate conclusions, but the magnitude of effects may vary. So what was interesting, when we had spontaneous deaths, it was not, in our studies, it was not immediately following the treatment. These mice died overnight when they're in their active phases. So we were looking during our day, or the mice's sleep cycle, because we did, that's sort of relevant to the clinical testing. And because we're not looking for seizures, we're looking for these more subtle measurements. So we did take 24-hour or 48-hour recordings. So we have the ability to go through and look at those data. And it's worth looking at all of it. But I think we'd be answering slightly different experimental questions if we did that. So, we chose in this data that I presented here, that hopefully will be out soon. We wanted to look at this window immediately after the chinic acid administration, so we could see the increase in sensitivity to treatments over time. But there's definitely a role for looking at 24-hour measurements and including that light-dark cycle in there as well.
Fantastic. Question here about the decrease in survival. Was there any indication of kindling that could have caused that the decrease in survival?
Well, I think that's what it is. Yeah. So there is definitely some deaths over time in any of these colonies. That's been known for a long time. Anyone that keeps colonies of any of these APP mutant animals knows that we do have, spontaneous seizures. But the fact that we had so many more in these mice after chinic acid treatment, we also see the same thing when we decrease vitamin C level on top of the APP PSM1 mice. We're more likely to use mice. And I think that is a cumulative effect of small seizure events, which is kindling, eventually leading to mortality. So yes, I do believe that that's what's happening here.
Fantastic. All right, next question here. What do you think is the most pathophysiologically accurate transgenic mouse model of Alzheimer's disease? I assume that it's kind of depends on what you're looking at, but what do you think about that?
Yeah, it 100% depends on what you're looking at. Yeah, that is a fantastic question. And if I could answer that question, I would be, far more advanced in life than I am now. So we are using this model knowing that it is an imperfect model, because it's a model and it's modeling the pathophysiology that we are interested in, which right now is this, slight altered EEG signal. And we're also interested, for example, in neuroinflammatory changes. And so we need plaques to exist so we can look at how microglia react during plaques. But that's not the same. Excuse me, that's not the same as saying we have a mouse with Alzheimer's disease. We're modeling one facet of disease. So, some people take the approach of putting in more mutations. So now we have tau pathology and amyloid pathology. Some people like to increase the mutation so you can see it in younger mice. Well, you could also argue that, Alzheimer's is a disease of aging. So if we're looking at these changes in mice that are not also undergoing normal age-related changes, is that relevant? Now we have a lot of new models with, TREM2 variants or, different gene changes that come from from the spontaneous Alzheimer's disease rather than the familial inherited Alzheimer's disease. I think you can choose any model you want that suits your purpose. You just have to be really acutely aware of the weaknesses of the model. And there are weaknesses in all of these models. We like to, I'll say, joke in the lab that all mouse models are wrong because they all synthesize vitamin C. And so the entire oxidative environment is different to the human brain. So we know that mice are not little humans. We just use them for the, for whatever it is we need out of them. So in this case, we wanted the altered EEG changes. And so any of the models that have that would probably work for these types of studies. But depending on what you're looking at, you may need to go with a different model. There is no good answer. It's a wonderful question. There's no good answer for that question.
Fantastic. Yeah, it's a great answer to a really difficult question. All right, so next question. All right, so thank you very much. Very interesting. I would like to ask about the absorption of vitamin C as part of the supplementation of mice, glucose, for example.
say that again. How the mice absorb it?
Yeah, about the
Glucose, for
absorption
example.
of vitamin C as a part of the supplementation.
I'm not quite sure I understand the question, but so in our gulo knockout mice, we know that the brain levels and the organ levels, correlate strongly with what we give them. So they do absorb them. There is a different transporter in the intestine, the SVCT1, that's responsible for uptake of vitamin C, and so we know that they do absorb it readily. You can max that out. You can replete those transporters, and so there's a limit to how much you can get into the mouse, but if you give it to them in the diet, they do absorb it. Most rodent models, guinea pigs are the exception, but most rodent models would not have vitamin C in their normal diet because you would not they do not need it. So I'm not sure about why glucose is glucose, for example. I'm not quite sure what that means. As far as I know, there's no interaction between glucose and vitamin C absorption because there is that specific transporter. So I'm not sure if that answers the question, but I hope so.
Fantastic. And I think in the interest of time, we'll just have one last question today. Do you think these data and studies like it mean that people should start taking anti-epileptics as a preventative treatment for Alzheimer's disease?
So, of course, it would be great to argue that there's a simple, answer like that. But, it's a really nuanced argument because, first of all, there are patients that undergo, sudden changes in cognition, and it's because of seizures. And when those are detected and you treat the seizures, they recover almost completely. So, yes, there are cases where that is the case. But in terms of preventative, it may be that there are people that benefit, but we do not understand enough to know who those people would be. We need to understand who would benefit from these. And then what are the correct types of anti-epileptics that would be beneficial? Because, of course, we also have side effects. We don't want to be, constantly treating people with every drug we have. So, yes, I think in future there may be subgroups that benefit from some types of anti-epileptic medication. Right now, we certainly can't just start throwing that at people and hoping for the best.
All right, fantastic. Well, thanks so much, Fiona, for the really great insights today. It's been a real pleasure having you with us.
Thank you so much, Liam. Thank you for inviting me.
Definitely. And a big thanks to the audience also for joining us for the event. And last but certainly not least, we'd like to thank DSI for sponsoring it. And so in closing, thanks again for taking part in this Inside Scientific webinar, and we look forward to having you with us again soon. Have a great day, everyone.